Student, in this session we are going to see about transfer RNA or tRNA. Before going to the transfer RNA, let us see what is RNA. RNA is nothing but ribonucleic acid. These RNAs are seen in the cytoplasm. When you see the structure of RNA, they are the polymers of ribonucleotides. What is this ribonucleotide? It is a combination of a sugar molecule, a phosphate group and a nitrogenous base. In case of RNA, the sugar molecule is ribose and the nitrogenous base includes adenine, guanine, cytosine and uracil. The most important function of the RNA is it is required for the protein synthesis. Then this RNA are classified into three types, namely mRNA, tRNA and rRNA. What is this mRNA? mRNA is the messenger RNA, tRNA is the transfer RNA and rRNA is the ribosomal RNA. All these three RNAs differs from each other by its size, formation and by its stability. Moving on to the session, that is transfer RNA. When you see the total cell content, the transfer RNA will be of about 10 to 15%. It has a molecular weight, that is low molecular weight of about 25,000 to 30,000 dalton. When you see the tRNA sequences, it consists of about 50 to 100 nucleotides. It is a single stranded molecule. This tRNA will also contain the modified or unusual bases. The modified or the unusual bases which is seen in the transfer RNA is ribothiamine, dihydroxyuridine and pidouridine. What is this ribothiamine? All these three modified nucleic acids are obtained from the nucleotides uracil. See, uracil undergo modification and it produces the unusual nucleotide, that is ribothiamine, dihydroxyuridine and pidouridine. In the ribose thymine, we have a methyl group CH3 here and a sugar molecule ribose. That's the name it becomes a ribose thymidine. Moving on to the structural aspects of transfer RNA. Transfer RNA is classified into three types, namely primary structure, secondary structure and tertiary structure. This primary structure will have a linear-like sequences and the secondary structure it is similar to the clover leaf and it is considered or it is named as clover leaf model, and the tertiary structure will be a three-dimensional structure which is seen to be in L shape. Here, this is the primary structure of transfer RNA. In the transfer RNA, there are four important arms present. The first arm is the acceptor arm and the next is the T arm, T side arm and anticodon arm. In the primary structure, since it is a linear structure, it consists of the 3' and the 5'N. To the 3'N, we have the acceptor arm to which the upcoming amino acids are accepted, followed by the T side arm and anticodon arm and DHU arm. DHU arm is nothing but which is rich in the modified nucleotide dihydroxyuridine. Moving on to the secondary structure of transfer RNA. This secondary structure of transfer RNA are more or less similar to the clover leaf and it is named as clover leaf model. This is the picture which showing the clover leaf. When you see the outline, both the transfer RNA and this leaf are similar, which has the three parts. This structure was first proposed by Robert Huiley in 1968. See, this is the structure of transfer RNA. In the transfer RNA, it consists of four arms, namely the acceptor arm, T side arm, anticodon arm and D arm. In the acceptor arm, at the terminus, it consists of AACC, that is the nucleotide adenine cytosine. This is the place where the amino acids get attached. And it contains an anticodon arm which recognizes the codon on the mRNA for the protein synthesis. This T side arm and T arm are rich in the unusual bases. In the T arm we have the dihydroxyuryl residues and T side arm is rich in pidouridine residues. Moving on to the tertiary structure of tRNA. The tertiary structure of tRNA will be a three-dimensional structure which will be in the L shape. If you see this in the view, the shape is in the L shape. This L shape is done or formed by the hydrogen bonding. Thus, we have seen the what is tRNA and its structural basis. Moving on to the function of tRNA, the most important function of transfer RNA, it acts as a carrier for amino acid to the site of protein synthesis. Since we have 20 standard amino acid, each amino acid will have a individual transfer RNA. Thank you.